our second video on genetics. In this video, we're going to look at things that don't really follow Mendelian genetics, what we just learned about. So we're going to look at some scenarios that don't really follow those predicted ratios that I just had you learn. So this is non-Mendelian genetics. And so I want you to take a look, and I'm going to zoom in on each of these three pieces of data. And I want you to pause your video at each one, and I want you to think, does this follow the ratios we discussed? Does this follow the logic we discussed earlier this week with Mendel? And if it doesn't, think about how we could explain the following data. And then as we move through this video and as we discuss all of this in class, we're going to start to see how all of this data looks really weird right now, but we do have phenomenon that can explain how we get this data. We're still in big idea 3, storage and transmittance of information, and that we do have inheritance patterns, but then that there are some that simply cannot be explained by Mendelian genetics. And so I want you to think about two things. Is we are going to do some quantitative analysis, and you will be asked to do a lot of that on the AP test in May. And what we're going to be doing is we're going to be looking at data, and we're going to see how the observed phenotype ratios differ very much so from the predicted ratios if we were to use pure basic probability and Mendelian logic. So we're going to see that there are 3 types of dominance. Now I just want to start off by sort of separating out the 3 in your mind. The first, complete dominance, you already know what this is. This is Mendel dominant recessive. So one allele is completely dominant over the other. The predicted ratios apply. So round is dominant over wrinkled. Okay, so that's complete. Now, the other 2 we're going to start to discuss today. Incomplete dominance is sort of an intermediate of the 3. So when we have a heterozygote, that heterozygote is going to display an intermediate phenotype. We had a red flower, and notice the difference in notation now, okay? Because we're not using Mendel anymore, we've got difference notation. So red flower crossed with a white flower, and what do we get in the heterozygote? We're going to get a pink flower. So that is incomplete dominance. Codominance is the last one. This is when both alleles are displayed equally in the heterozygote. So if we think of the same cross where we have the CRCW, what we get is actually patches of red and white in this heterozygote. So that's codominance. And we're going to take a closer look at these last 2 in this video right now. So incomplete dominance, I have 2 practice problems for incomplete dominance for you to do really quickly. Just keep in mind that, remember that whenever we have the heterozygote, it is going to be this intermediate phenotype. So this first practice problem, we're looking at guinea pigs, and we have the cream color is this heterozygote. So the cream is sort of the intermediate between the yellow and between the white. And they're going to have you do a cross, and they're going to have you tell me what ratios does that produce. And here's a second one, and this is with the chickens I was telling you about. So go ahead and do these practice problems in your notes. Okay, the last one was this codominance. This is also called multiple alleles. Now, so far, I'm reading up here, we've only examined traits with 2 traits, right? With, wrinkled versus round, yellow versus green. And this is easy to visualize because we know that a diploid organism has these 2 alleles, one from mom and one from dad. However, that's not always the case. Sometimes more than 2 alleles can exist, although despite that, any given individual still is only going to have 2 alleles. It's just in a population, there are going to be multiple ones that exist. And the classic example, the only one we're going to look at, is blood groups. And so what I want you to write down in your notes right now is this chart right here. I need you to know the notation for the genotype and then what phenotype that notation is going to tell us that it is. Now, we will go over more in class where these come from. They actually come from little carbohydrates that are on the cell surface of the red blood cell. And they're either a B carbohydrate or an A carbohydrate, or in the case of the AB, it has both carbohydrates on the surface. And we'll talk about what that means as far as donation and receiving of blood. So now that you know those notations, you should be able to do these practice problems and do these 2 in your notes. Okay, another sort of funny scenario that cannot be explained by simple Mendelian genetics is polygenic inheritance. So this refers to inheritance of a trait that is attributable to 2 or more genes. And this can be measured quantitatively And it varies along a continuous gradient that we often depict by a bell curve. So I'm going to show that to you real quickly. I'll come back to that slide. So the best example is skin color. So you can see this is the bell color of skin color, or the bell curve of skin color. Skin color is due to many different alleles that combine together to show this sort of curve in gradation of color. Here's your homework, can you come up with one other example of polygenic inheritance and 2 examples of diseases that would be considered multifactorial? So let's go back. Multifactorial simply means that yes, it's polygenic, but there's an additional component to it, and that's the interaction of the organism or of the genome with the environment. And we're going to continue to look at this throughout the year, how closely we are connected with our environment and how environmental factors can really change our DNA. It's kind of crazy. So come up with 2 examples of diseases that are considered multifactorial. Okay, now here is a really, really cool phenomenon. This is called non-nuclear inheritance, also called cytoplasmic inheritance. So when we think about DNA, we automatically think of the nucleus, right? We think about inheritance, we talked about mitosis and meiosis, and the chromosomes are the nucleus, and they're separating, and they're going to form the gametes for the daughter cells or what have you. But think about the rest of the cell. There's a lot of other stuff in the cell. There's organelles, especially mitochondria, and in plants, mitochondria and chloroplasts. Now, earlier in the year when we talked about the cell, we briefly touched upon the fact that these 2 organelles sort of have an evolutionary history, don't they? That they were at one time, we believe, their own prokaryotic cells because they have their own ribosomes, they have a double membrane, and they have their own DNA, don't they? Because of that, there is inheritance when you receive a mitochondria from a parent or when you receive a chloroplast from a parent if you're a plant. But they don't really follow basic mitosis or meiosis rules. It's not really a neat separation or segregation into the daughter cells. So they are actually randomly assorted into gametes and daughter cells. So because of that, they absolutely do not follow any rules whatsoever. And as I was researching this more, I mean, the data out there is just so off the wall. There's just no rhyme or reason for how chloroplasts or mitochondria get passed on to daughter cells. So we're just going to take a brief look at 2 scenarios here. The first is the mitochondrial DNA. So this is transmitted by the egg and not by the sperm. So this is because the egg is sitting there and it's full of the cytoplasm and it's waiting to be fertilized. So that has the mitochondria in it. The sperm is very tiny. It does not contain any mitochondria. So any mitochondrial determined traits are maternally inherited. So if your father had any mitochondrial diseases, you would not receive them. You would only receive them from your mother. So I want you to do a little bit outside research and name 2 mitochondrial inherited human diseases. So let's take a look at what this looks like in a figure. So here we have, over here, we have an affected father who has this mitochondrial disease and an unaffected mother. And as I just said, all the children are going to be unaffected. On the other side, though, we have an affected mother and an unaffected father, and unfortunately, all the children are going to be affected because that disease is carried from the mother in the egg on that mitochondria. Very interesting. Okay, now over here on the right, we have an even more interesting scenario with plants. And these plants are called 4 o'clock plants. And you don't have to know all the details of this, but what they did was they crossed flowers from white, green, and variegated, which is just sort of a combination. This is variegated in all sorts of combinations. And so this is sort of the female over here on the left, and then over here on the top, this is the male. This is the pollen grain. And what they found over here in the conclusion down here, it is determined from where the female seed originated. So same idea. The mother plant, if you will, is going to provide that chloroplast DNA. The pollen grain, again, so tiny, it does not have any chloroplast in it. So that color must be on the DNA that's in the chloroplast, and that is provided by the mother seed. So as you can see, it always matches the female parent. In this case, we'll talk about in class, but I want you to think about why, with this case, you would get all 3 types. So think about that. And if you would like, I did put down here at the bottom, if you can see it, this website down here. It's a really short read. It might take you 5 to 8 minutes to read, but it's really fascinating information about non-nuclear inheritance. Okay, almost done. We have to talk briefly about sex-linked inheritance. This is also called X-linked inheritance, and it's called X-linked inheritance because sex-linked genes reside on the X chromosome. And why? When we look at the X chromosome over here on the left, it's huge. There is so much genetic information on there. So any genes that we look at are going to be on the X chromosome. The Y chromosome is much smaller and does not have as many genes. And what's really interesting in that is that both in mammals and in fruit flies, which is the model organism for studying sex-linked diseases, it is the same. It's the big X and the small y that determines sex. So again, very short piece of homework. So let's take a look about notation. So as we do these sex-linked problems, students mostly struggle with how to actually write it out. So you get the concept, but writing it out can be confusing. So females are XX and males are XY. Again, that's the same in mammals and in flies. Because of that, because a male only gets one X, okay, they're XY, and this is a key concept here. They only have one X. So if that one X has the disease allele on it, they're out of luck. They express the disease. So X-linked recessive traits are always expressed in males. They're sometimes expressed in females if the female receives both recessive X chromosomes. Okay, so how do we notate it? We use X and Y with superscripts. So for example, X big R and X little r would be an example. And then when we look at fruit flies, I gave you some notation here. The wild type is also known as the normal phenotype, no mutation. And we notate that, and this is weird, we notate it with the mutant, the first letter of the mutant. So the mutant is white, so that would be W, and then a plus sign. Now, the mutant is just the W. Okay, so again, the wild type is the W with the plus sign. Mutant is just the W. Again, let's look at wings. Vestigial wings is the mutant, so the mutant is just VG, and then the wild type, the normal wings, is VG plus. I know, very strange. You just have to get used to it. So Thomas Hunt Morgan was in the early 1900s. Did some really great work with fruit flies and really basically studied and founded the idea of sex-linked inheritance. And so we're going to take a look really briefly at these two scenarios. So when we look at this one, we have a red-eyed female and we have a white-eyed male. He crossed these, and what he found was that half of the offspring were red-eyed females and half were red-eyed males. But then what did he do, right? For the F1, he self-fertilized. He mated the F1 generation to get the F2 generation. So now when we look at the F2 generation, we have the females. The females all have red eyes. And then half of the males, when we look at the males over here, half of them have red and half of them have white. Now, why is that? Because when we go back up here, this was a wide-eyed male. So one of the parents was a wide-eyed male, so it carried this little W on the X chromosome. When we look at the F1 female, that F1 female has red eyes, but it does carry that little W right there. It's a carrier of that wide-eyed allele. So then when those, law of segregation, when those two X's separate, half of the males are going to be out of luck again, and they're going to receive, looking down here, that little white. And remember, it only takes one little white allele to have white eyes, because that's a recessive trait, and males only get one X chromosome. Okay, I'll zoom in on this one for you, and you can take a look at this one. I won't explain it, but this is just the opposite. So this is a wide-eyed female. Again, she's got to have two little W's, though, to have white eyes. And Morgan crossed it with a red-eyed male, and you can follow through and see what happened there. I also have another example for you to look at. Again, looking at sex-linked, and this is on the X chromosome. So the mother, but this is actually looking at an X-linked dominant allele. Okay, so this is a little bit different scenario. So all it takes is one of those in the female to express it. So when these two separate, again, half the males, okay, so one half of the male's affected son is going to have that. And then again, half the daughters are going to have it because it's dominant. You only need one of those to express it. So you have to start to become very comfortable with looking at data and understanding whether or not it's dominant or recessive. And I've got one practice problem for you to look at for sex-linked traits. And lastly, I don't have time to discuss this, but go ahead and write down these notes, and we'll talk about this in class.